Okay, here we go. 7th grade life science DNA structure PowerPoint notes. Now we start this off with talking about a little bit of history of heredity. Now remember, heredity is the passing on of traits from a parent organism to the offspring. Now remember, we studied about Gregor Mendel. Gregor Mendel was the monk who in the 1800s discovered that traits were passed on from generation to generation. And he did that by studying pea plants and crossing two parents with two different traits, two plants with two different traits, and recorded the outcome. And he came up with his three main principles, the principle of dominant and recessive, and we studied all that before. But the thing about it was that he had no clue of how this happened. He didn't know if there was some sort of factor present in the organisms that controlled that. So he died not knowing how it happened. So the next person that kind of came into the history of figuring out the secret to life, or how traits are passed on from parent to offspring, was with Frederick Griffith. Now he performed a very famous experiment, and actually his experiment involved working on a vaccine for Streptococcus pneumoniae. Now this is a bacteria. Here's a genus species. Here's the genus. Here's the species. This is a species of bacteria that causes pneumonia. Now it is in the genus Streptococcus, which there's another type of strep that causes strep throat, but this is one that caused pneumonia. And he discovered that there are two strains of this bacteria. And he looked at it under a microscope. He looked at it under a microscope, the two strains. One of them had a smooth capsule on the outside. The other one had a rough capsule. Now, it turns out that the smooth capsule organism, little bacteria, caused the pneumonia disease, whereas the rough capsule one did not. Okay, so he kind of thought to himself, well, how does this work? So he went to his lab and he had some mice or lab rats that he worked with. And in this experiment, he took the live, living, smooth bacteria, the ones that actually caused the disease, injected it into some mice, and lo and behold, the mice died from the disease, obviously. Then he did the same experiment, only he used living rough bacteria, the ones that didn't cause the disease. And, as expected, these mice lived. So for his third experiment, he took some of the dead, smooth bacteria. So he killed the bacteria, but he didn't destroy them. Okay, so all the parts were there. They were just not living bacteria any longer. Injected the mice with that, and they lived. Okay, so apparently these bacteria needed to be alive to do the killing. It's kind of like if you have a gun sitting there, the gun can't kill unless a living thing comes and does it. Okay, same thing here with these bacteria. So the fourth part of his experiment was he took some of these live rough bacteria, the harmless ones that are living. He mixed them with the dead smooth bacteria, okay? And the mice died. So what came out of this experiment was that there is something that actually controls traits, like being able to cause a deadly disease. So his famous experiment proved that somehow the dead bacteria have passed their disease-causing substance to the harmless living bacteria. So the main thing that came out was Griffith discovered a factor that could transform harmless bacteria into disease-causing bacteria. So yes, there is a factor, and this experiment was called Griffith's transformation experiment. Okay, so the next question is, okay, we know it's a substance that causes traits to be able to be passed down. So what is this substance? Well, now we know it's DNA. So Oswald Avery, who was born in Nova Scotia, came to the United States, and he was a microbiologist, a bacteriologist also. And he determined that the actual factor that Griffith discovered was DNA. So he and his team determined that genes are composed of DNA. Genes are what are transferred from one organism to another that controls certain traits. And he discovered that they are actually the molecule DNA. So now we know there's a factor. Now we know that factor is DNA. Okay, so Avery, by the way, was recognized a lot for his work, but he never did win a Nobel Prize. And actually what they did kind of to honor him is he actually has a crater on the moon, a lunar crater, that's named after him. It's called the Avery crater. That's just a little FYI there. All right, so now we know it's DNA. Okay, then this next guy, Erwin Chargaff, he discovered that DNA is made up of some bases. Okay, it's made up of some bases. These are molecules, and they discovered, he and his team discovered, that there's the same amount of adenosine as thymine in a molecule of DNA, and the same amount of cytosine as guanine. So therefore, and we abbreviate these bases as adenine is A, thymine is T, guanine is G, cytosine is C. So there are equal amounts of A and T and G and C. All right, so what that did, okay, we know it's DNA. We know some facts about it, like the bases. There's equal amount of A and T and G and C. Then came the big race that we saw the video about Photo 51. It was kind of like this mad race that whoever finally discovers what the actual structure of DNA is going to discover the secret of life. Because once you discover what it looks like, you can figure out how it works, okay, which is very important. So remember Rosalind Franklin, they called her the Rosie or the Dark Lady and all that. And her lab partner Wilkins used that X-ray crystallography, and they discovered that DNA was actually a double helix through this photo. By looking at this photo, they discovered that it's a helix, and it's a double helix. And they were in competition with Watson and Crick, who used model building. So they kind of stole Franklin's Photo 51 to help put together the correct model. So they credited Wilkins and Franklin, sort of. Wilkins also got a Nobel Prize, but Franklin did not. So Rosalind had passed away, and the Nobels are not awarded posthumously. So once you have died, you cannot be awarded a Nobel Prize. So we know kind of the real story behind that. We know that James Watson actually wrote the book, The Double Helix, describing how it was all discovered and that sort of thing. And he's actually still alive. He's 84 years old, or 85 now, maybe. He was born in 1928. And he's actually still working and writing papers. Francis Crick died, I think he was 77. He died in 2004, but they are the ones that got the Nobel Prize for actually discovering the actual structure of DNA. So, let's talk about the actual structure of DNA. It actually is a nucleic acid. We've talked about nucleic acids before as one of the 4 types of organic compounds. Remember, you have carbohydrates, you have proteins, you have lipids, and then the important nucleic acid. So a nucleic acid, review, old enzyme means old acquaintances. Nucleic acids contain the 5 major, the CHNOP, 5 major elements in living things. They are made up of nucleotide monomers. So, a monomer, actually, of a nucleic acid is called a nucleotide. They store genetic information. They help make proteins. And examples are DNA and RNA. So what does DNA stand for? Deoxyribonucleic acid. You'll need to know that. Deoxyribonucleic acids. So these are 5 bullet points that go at the bottom of your notes on the first page for nucleic acids. All right. So again, it stands for deoxyribonucleic acid. Okay. So turn the page. We're going to talk real quickly about what is the monomer. What is a nucleotide? A nucleotide has 3 major parts. So we got A, B, and C here. The 3 major parts. You have a phosphate group. You have a sugar, and that sugar actually is deoxyribose. And then you have a base, an N base we call it. So you have a phosphate, a sugar, and a nitrogen base. Okay, we're not going to worry about the 5' ends and all that sort of thing. So there's the phosphate. There's the sugar. Now next to sugar, I want you to write deoxyribose. That is the sugar. See how it ends in "-ose"? When we studied carbohydrates and simple sugars, they end in "-ose". So in DNA, the sugar is deoxyribose. In RNA, another kind of nucleic acid, it's ribose. So the sugar is this 5-carbon sugar here. Each point is a carbon. So there's the phosphate group, there's the sugar, and then we have the nitrogen base. Now there are 4 nitrogen bases. Okay, there are 4 nitrogen bases. There's adenine. This is what Chargaff discovered. Adenine, guanine, thymine, and cytosine. So those are the 4 bases. You don't need to know about purines and pyrimidines at this point. Okay, so base pairing. When bases pair or get together, as Chargaff figured out, adenine always binds to thymine, so it's A and T. Guanine always bonds with cytosine, G to C. Okay, one way that I've always remembered this, A to T and G to C, is totally awesome and groovy cool. Totally awesome, groovy cool. Okay, so they bind together with hydrogen bonds in between. Okay, so let's take the nucleotides and bind them together the way they are in DNA. Okay, so here's a nucleotide. Okay, you have the phosphate, you have the sugar, deoxyribose, and then you have an N base. This one happens to be guanine here. Okay, so let's think about the rule. Totally awesome, A to T, G to C. So what is this base here going to be? It's going to be C, cytosine. and here's the sugar, and here's the phosphate. So when we put together the nucleotides to make the DNA molecule, you have to think about base pairing, G to C. Okay, so the complementary nucleotide pairing here would be C. There it would be G, and we go on down to the other nucleotides, and they pair up together. Okay, so here again is going to be another nucleotide with the base, the sugar, and the phosphate. Here's another one, the base, the sugar, and the phosphate. And the phosphates connect the nucleotides together on this side of the molecule. And in the middle, the hydrogen bonds between the base pairs. And then again on this side, the phosphates hold the nucleotides together there. So here is a section of DNA. It's a double helix. Here's one side of the strand. Here's the other. They're held together by the hydrogen bonds between the bases. Don't worry about the 5' end or 3' end and all that. So it's ladder shaped. Okay? It's ladder shaped. You have the side chains of the sugar and the phosphates. Okay, those are like the sides of the ladder. And the rungs of the ladder here are the base pairs. Okay, now we call it a helix. Why do we call it a helix? Because it's twisted. Okay, so why do we have DNA? What's the purpose of DNA? Well, it contains the genetic code. What does it code for? It codes for proteins. So it pretty much tells you actually what makes you look the way you do. Okay, so it's DNA and proteins. Okay, real quickly we're going to talk about RNA. RNA stands for ribonucleic acid. It's a helper molecule for DNA. In other words, it helps interpret the DNA code for traits. The difference between DNA and RNA is single-stranded. That's one difference. It's made up of nucleotides as well, just like DNA, except the N bases, the nitrogen bases, are a little bit different. The sugar is ribose. You do have phosphates, and you have the nitrogen bases. Okay, just like in DNA. Again, A is the sugar, B is the phosphate, C is the nitrogen base. Okay, so let's do this, only it's single-stranded. You have to remember that too. There's the sugar, which is ribose. There's the phosphate. Here is the nitrogen bases. Okay, now the base pairs are a little bit different with RNA. They do have guanine and cytosine pairing together. Groovy cool, G to C. Okay, however, there is no thymine in RNA. So in this case, adenine pairs with a nitrogen base called uracil. So it's A to U in this case with RNA. Okay, so here we go. Rules for base pairing. See, with RNA, it's like that. We'll go over more of that later. Okay, so that is all we're going to, well, let's go over comparing DNA and RNA. The sugar is deoxyribose, so you're going to check DNA. The sugar is ribose. You're going to check RNA. Adenine base is present. Well, that's in both of them. Cytosine is present. That's in both of them. Guanine is present. That's in both. What about thymine? Well, that's only present in DNA. Instead of thymine, RNA has uracil. It's a double helix. That's only DNA. It's single-stranded. In other words, it only has one side. That is RNA. It's in the nucleus. That's both. And the cytoplasm. Okay, here we go with something new. DNA never enters the cytoplasm. It is always in the nucleus. Okay, RNA does both, and we'll talk more how that works later. Okay, DNA stores genetic information. Functions in protein synthesis, that's both. Composed of nucleotides, that's both. It's used as a template for synthesis of proteins. We'll talk about that later. That's DNA. Transcribes the template. We'll talk about that later. That's RNA. More than one type. There's more than one type of RNA, and we will talk about that later. So mostly what I wanted you to get out of this PowerPoint today here is the actual structure of DNA with the nucleotides.